Okay, so welcome everyone. I will start to say a few words as people are trickling in. Um, it's my pleasure to be the chair of today's webinar, and I'll share my screen for the program. Yeah, so my name is Laura Wisse. I'm associate professor at Lund University. And I have had the pleasure to work with Ricardo Insausti for a very long time. And I'm glad that we were able to pull this webinar together to remember him as the wonderful person that he was, but also to pay tribute to the amazing science that he left behind for us. Um, so yeah, actually, I'm not the only chair. The other chair is Mar. Would you like to introduce yourself, Mar?
Hello, everybody. Yes, for me it's an honor to welcome to this session, and I just want to grateful to Laura because she is the aim of this session to tribute to Ricardo Insausti, which is our point of union in this session.
Thank you. So the program you can see here. So we'll start with two parts of Ricardo's work on anatomy in non-humans and in humans. We'll have a little bit of a break, and then we'll go into Ricardo's two projects in which Ricardo was involved a lot that were also more recent. And then we'll hear from some of his students and friends and family. And hopefully in the end, there's some time for questions or other inputs. Unfortunately, I don't think we'll have a lot of time for questions on the talks, but we'll see if we can manage to make a little bit of time for it. Before we start, I also wanted to mention that this webinar is recorded, and we'll put it on YouTube afterwards, but we'll probably only put the four talks on YouTube. And finally, I would like to ask you to keep yourself on mute until the time we can ask questions and so on. All right, so then I would like to introduce our first speaker, Professor Menno Witter from the Emeritus Professor Menno Witter from the Kavli Institute of Neuroscience in Trondheim. And he will present the Insausti perspective on structural organization of the hippocampal system in non-humans.
Thank you very much, Laura, for this introduction. And so, first of all, Laura, thank you for putting this together with Mar. I think this is a very appropriate occasion to think about Ricardo and tell a little bit about his work. And I have decided to talk a little bit on how I got to know Ricardo and how we started to work together and what kind of things I learned from Ricardo. And that's what I call the Insausti perspective. And I mainly will focus on the work on non-humans, and then Monica will take over, focusing more on the human work. So Ricardo and I actually met, I think it was in 1985 or 86. I tried to recall it. I couldn't find the real date, but I think we first met at the Salk Institute when Ricardo was working for already a few years with David Emerald, who was then working with Max Cowan. Later on, when Max moved back to Washington University, David took over the lab and became the director. And both Ricardo and I stayed working and meeting both at the Salk Institute, but I also started to meet more and more often in Spain with Ricardo. So here you see this image of the Salk Institute, which to me still looks like the image of a temple of science. It's almost, this is a very amazing building on the coast of the Pacific. And so Ricardo started, I think, a couple of years before I first started to work with David. They published their first paper on the monkey entorhinal cortex of an input, as you see in the title, in 83. So that was the the first move from Ricardo from doing his thesis work on the trigeminal nerve and moving into the hippocampal system and the memory system. So when we started to interact, I think the question was, just as a background, what did we actually knew? What was our knowledge at the time when we started to work together? And so we all knew about the concept of a limbic lobe, a set of structures in the brain that was described by Broca. I'll show a slide in a minute. There was a limbic circuit underlying emotion. There was a tape circuit, and then we learned about the hippocampus as a main component of the memory circuit. And then we learned more and more about the entorhinal cortex. So let me start with the limbic lobe. This is a picture of the brain, of a mid-sagittal view of the brain of an otter. And it was Broca who described this ring of structures around the corpus callosum here. And this ring of structures, cortical structures, together with the olfactory bulb and the olfactory peduncle, he named the grand lobe limbic because it is the border, the limit of the cerebral cortex attaching to the diencephalon, to the thalamus, which is dissected here. So if you take this image and you move that into a human brain or a monkey brain, I took the human here, you see that you can recognize the cingulate gyrus and the isthmus that connects it to the parahippocampal gyrus, and that more or less represents the limbic lobe in the human. And the limbic lobe was, as a pure anatomical term, Broca did not really talk about functions. He just named it as a structural entity. It got its first real functional connotation by the work of Pepys or Papez, depending on how you want to pronounce his name, who argued that there is this set of structures of the hippocampus having a fornix connection as its main connection, moving into the memory bodies. And then from the memory bodies, you had the mammalothalamic tract into the anterior thalamus. And here in the cartoon-like representation, Pepys suggested that this part of the thalamus would be a main input to the cingulate gyrus, including the spinal cortex, that then would have connections by way of the cingulate, the fiber bundle, into the parahippocampal structure that would feed into the hippocampus. So Pepys suggested that there was a kind of closed loop in the brain that he suggested would be underlying emotional functions in the brain. So this was, at the time, was a pretty well-established concept, but that changed when Scoville and Milner published their paper in 1957, where they reported on bilateral hippocampal resection in HM and hemolysin, as we now know. And if you look at his brain and compare it to a control brain, and I zoom in a little bit, there we go, then you see that what in a healthy brain looks like this quite smooth surface of the ungual portion of the medial temporal lobe. So this is a ventral view of the human brain. And here, of course, you're all familiar with it. You see the lesion in HM, where on both sides, a large part of this cortex and the underlying hippocampus was removed. And that resulted in a loss of memory, particularly newly formed memories. So there was this idea that these structures were critical to store or at least build new memories, not so much store them, because HM still had remote memories from before the surgery, although they were not as good and as detailed. So the idea was that particularly the hippocampus might be a critical structure. So the structures that were removed included the hippocampus, a large chunk, about the anterior 2/3 of the hippocampus, and then part of the amygdala and the entorhinal cortex were removed. So either of those three structures or the three structures together would be important for memory. And that very quickly led to this idea, going back to, oh, the fornix is the main output of the hippocampus into the memory body. So probably the fornix would be as crucial as the hippocampus for memory. Unfortunately, it turned out that people try to mimic the symptoms of HM by doing fornix lesions in animals, because the idea was that it was a lot easier to cut the fornix than take out the entire hippocampus in an animal. But it turned out that fornix lesions never ever mimic the full-blown or full-scale deficits that HM showed. So there was a kind of problem that we didn't know how the hippocampus did the job that it was supposed to be doing, storing, consolidating memories, but eventually not storing them as a final storage, because as I said, if you remove the hippocampus as is in HM, you still have memories from before the damage. So then the story developed because a series of papers from groups at Harvard and MIT published that there was a connection from the hippocampus directly into the entorhinal cortex. And this is an image taken from this paper here in Science. It was collaborations with the people of Deepandia, Gary Van Hoosen, Doug Rosine. They all worked on this project together, and they showed by doing an anatomical tracing experiment that if you inject and make an injection of tritiated amino acids in the medial temporal lobe in the hippocampus, you get a beautiful labeling of terminals, of axonal terminals in the deep layers of the entorhinal cortex. So based on that idea and that finding, they proposed that there probably will be an output pathway of the hippocampus mediated by the entorhinal cortex. And just to show you in an image that this is really the case, this is a voltage-sensitive dye experiment that I did many, many years ago in Sendai, where I still hold a visiting professorship, where you looked, if you drive the dentate gyrus by electrical stimulation, you get a beautiful volley of activation traveling into the entorhinal cortex and then into the adjacent cortex. So it really shows that this is a major output for the hippocampus to communicate with the cortex. So that triggered a lot of work that Ricardo has been contributing massively to try to understand how this whole circuit was wired up. So to start with a cartoon, at the time of the study of HM, this was what we approximately knew about the hippocampal system and the entorhinal cortex. It was well known based on the work of Cajal that the entorhinal cortex had a major input into the hippocampus, particularly hitting the dentate and CA3. Then there was a series of sequential projections inside of the hippocampus, and then both CA3 and CA1 would project out through the fornix. Now, the fornix as a main output was discarded, as I just argued before, and that's wrong, actually. Nowadays, the fornix has come back into the field of interest of memory, but at some point, people decided that the fornix wasn't relevant. It was actually the output of CA1 and subiculum to the deep layers of the entorhinal cortex. And in addition, it was shown by many different studies that the entorhinal cortex had a more complex input system, that there were also projections to CA1 and to the subiculum. And we knew, already partially based on the work of Cajal, but there was more and more evidence added to it, that, oops, I need to hit the right button, that the inputs from the cortex mainly hit layers 2 and layer 3 of the entorhinal cortex that would provide the input to the hippocampus. And then the hippocampal output would reach layer 5 of the entorhinal cortex. It would, in turn, neurons in layer 5 would project to the cortex. And then there are connections, both at the level of the intrinsic connectivity of the entorhinal cortex, as well as through entorhinal cortical interactions, that you could have processes going on like reverberation and consolidation. I don't have time to go into the details, but this was the real scheme that Ricardo and I started to work on, focusing on the question, what is the contribution of the entorhinal cortex, and how can we understand this better? So I have to go back a little bit again in history, because in order to understand the entorhinal cortex, you need to figure out how to describe what you're doing experimentally. And in that process of coming up with good descriptions on which you could base your experiments and describe the results of your experiments, Ricardo and I started, together with many, many other colleagues, to focus on how do we give names to structures. Of course, there was a long history, and based on that history, we the field more or less accepted that the entorhinal cortex could be subdivided in two main parts, the lateral and medial entorhinal cortex, color-coded here in the rodent, in the rat in this case, in the medial entorhinal cortex in green, and in yellowish, goldish, the lateral entorhinal cortex. There was a concept that there could have been an intermediate third area. Lorenzo Renaud, who's considered to be the godfather of the entorhinal cortex in terms of its structural organization, suggested that there might be an intermediate zone, but Lorenzo Renaud actually, in his 33 and 34 paper, decided that all these subdivisions were more or less irrelevant. The entorhinal cortex was an entity that was important because it provided an input to the hippocampus. But most of the rodent work was focusing into the subdivision into two. But as I said, there were other subdivisional schemes. And I think that the work of Ricardo, together with David Emerald, firmly established a subdivision in the monkey where they proposed 7 different subdivisions. Now, this is their subdivision of the entorhinal cortex. There's some adjacent parahippocampal areas that I won't have time to go into. But if you compare this map with a more simple map where you have a medial and a lateral entorhinal cortex and an intermediate, this was the description that Van Hoosen and Pandya and Rosine used for their monkey studies. So it's 2 or 3 subdivisions. Rose came up with something like 18. Skonina had a similar 13 type of subdivisions. So the field was really not agreeing on how many subdivisions there are, but I think that Ricardo contributed immensely in establishing a useful format that allowed many people to do both anatomical experiments in the monkey, but also to do physiological experiments and behavioral experiments, lesion experiments, and to describe properly which part of the system are we talking about. And in the same year, actually in the same volume of the Journal of Comparative Neurology, they published 2 influential papers on cortical and subcortical efferents in the monkey. And I think that these papers, together with this pseudoarchitectonic description, move the field forward into almost like a tsunami of publications on connectivity of the entorhinal cortex in relation to the connections with the hippocampus. And so since Ricardo established this important subdivision in the monkey, he and I agreed to work together and try to see whether a similar, more complicated subdivision would be useful in the rodent. So we published a paper together in 97, and this is a cartoon, and I'll blow up the cartoon a little bit, where we suggested that there were 6 subdivisions in the entorhinal cortex. And the entorhinal cortex, which is important to mention here, was defined by the cortical domain that had direct projections into the dentate gyrus. So that was our definition of deciding which part of the cortex is entorhinal and which is not. And we eventually had 6 subdivisions, as you can see here, being as closely associated to the 7 subdivisions in the monkey as we dared to do. But later on, we had to take this one out, the amygdaloid entorhinal transition, because it turned out, based on beautiful work done by Asla Pitkanen, and I'll show an example of that a little bit later, she showed that this area does not project to the dentate. So later on, we removed that area, ended up with 5 subdivisions in the rodent entorhinal cortex. And we published papers together on all these different subdivisions, how they are related to different cortical fields and subcortical areas. So it helped us to make comparison between the monkey detailed studies and the rodent detailed studies. And just to end up, this is the only human slide that I'll show about Ricardo's work. In the human, he decided, together with David Emerald, to subdivide the entorhinal cortex into 8 subdivisions. They added this particular subdivision, which is now also known as the cortex that makes the surface of the ambient gyrus. And this was an area that was originally referred to by Brodmann as an area that looked like the entorhinal cortex, but Brodmann, who gave numbers to the cortex, called all of this area 28, and this was area 34. At least that's the most likely homology in terminologies that we can come up with. And just to summarize the number of subdivisions in the human entorhinal cortex, as I did in the monkey entorhinal cortex, as you can see, there's many different ways of subdividing the human entorhinal cortex, varying from 2 to 3 subdivisions, lateral, medial, and an intermediate, up to 23 fields, as Rose suggested. So the question that comes up, and I'll return to that at the end, is what is a useful number of subdivisions? You have to be pragmatic. You can continue to divide all the way up to a neuron. And Ricardo and I had endless discussions on how many subdivisions do we need to handle our data. And I think that the choice for 8 subdivisions in human is useful and practical, although nowadays, with the current genetic and spatial transcriptomic data, we might have to change that view a little bit. And Ricardo made a very important statement that I want to quote here, that we still don't know. This was published in 95, but even now, we still don't know whether all these areas that we subdivide in different species are actually homologous or not. We think that there are homologies, but we're not sure. And I hope that the transcriptomic data, and particularly the spatial transcriptomic data, will eventually allow us to resolve that problem. Now, all the work that Ricardo did on the monkey after he left the peripheral nervous system, the trigeminal nerve of his PhD thesis, since 1995, he really started to focus more and more on the human, and you'll hear a lot more about that in the next talk by Monica Muñoz. But he published a few papers that were still not human. So he published this paper on the red connectivity of the hippocampal formation in the amygdala. And if you can read it carefully, there's the name of Asla Pietkanen, who helped Ricardo and me and convinced Ricardo and me that this area that we originally included in the entorhinal cortex as the amygdaloid entorhinal transition is not part of the entorhinal cortex because it does not project to the dentate gyrus. Then he published 2 papers on the baboon as a side work on the rhesus monkey work, but particularly as I considered as his step into moving more and more into the human work, because this is an MRI study. So he started to play with the tool that is used a lot more in human studies. And he started to take some data in the baboon as a possible model for Alzheimer's disease. So again, pointing towards his increasing interest in what can be done in the human. But at the same time, he continued to work and publish papers on the monkey. And here are just 2 other papers on the monkey entorhinal cortex. And the important, and that's the reason that I picked this paper, is the title of the paper, The Convergence of Unimodal and Polymodal Sensory Inputs to the Entorhinal Cortex, because this title is nowadays a core feature of what we think the entorhinal cortex is doing, namely bringing cortical information together and provide the hippocampus with a very complex representation of what our cortex knows about our external world as well as our internal representation of the world. And with that, I want to wrap up. I think that Ricardo strongly insisted on the relevance of having a precise structural and connectional basis that could function as a foundation for functional understanding and as a solid foundation to represent data from different labs, from different animals, into a coherent framework such that we all knew what we are talking about. And I think Ricardo was really a very precise person. He was a lovely colleague. I really enjoyed working with him. He always insisted that if you wanted to contribute to a scientific field, you had to be precise and you had to be as perfect as possible. If it wasn't perfect, he would push to make it better and better until he was finally convinced that it was good enough. He moved from his work on the peripheral nervous system through his work on the monkey and ended up with the last part of his career spending an enormous amount of time on the human hippocampal system. I really enjoyed traveling with him for a while, since the mid-80s. I worked with him in La Jolla, I visited him in Pamplona. I still vividly remember looking at the bullfights in the streets of Pamplona from this tiny balcony in the main street. I visited him many times in Albacete, and Ricardo has been extremely important for me to learn the Spanish neuroscience and neuroanatomical society. I made an enormous number of friends, and I have a very extensive network nowadays still in Spain, and I owe that to Ricardo. And for all of this, I think he will be remembered, and it's worth to remember him. And with that, I thank you for listening. Thank you.
Thank you, Menno, for this wonderful overview of his work. I guess we have 2 minutes. I wonder if someone, I guess it might be too short. Let's maybe move on to the next talk.
Yeah.
And in the meantime, if people want to put a question in the Q&A, we can see if we can take some afterwards. Yeah, thank you. All right, so then I will introduce our next speaker, who is Associate Professor Mónica Muñoz-López from the University of Castilla-La Mancha. And her title you can see here, The Legacy of Ricardo Insausti, Structural and Functional Organization of the Hippocampal Formation.
Thank you so much, Laura, for organizing this event. I think it's a beautiful idea. And I'm only honored to be part of this panel of speakers who are amazing researchers. And it's for me an honor to have been working with Ricardo for such a long time. And to think about him just now. So let's start. I think it's difficult to think about Ricardo and not to think of one of his beautiful dissections of the brain. And this is just a photograph of him together with the focus of his career that has been the entorhinal cortex among the medial temporal lobe areas. This is just the center of the medial temporal cortex, the entorhinal cortex, but he also did this beautiful work dissecting pictures that show in a three-dimensional way where the hippocampal formation is in the human brain. Perhaps it's difficult to recognize all the structures that have been cut here, but you can see here the encephalum around here, and the hippocampus and the entorhinal cortex just there, and all the fusiform gyres on the side. But also, it's difficult to think about Ricardo without remembering how many hours one spends with him in the microscope looking at all these areas of the hippocampal formation. Just browsing with the microscope area after area, the cytoarchitectonics of all these subdivisions. And I left this picture completely naked because I'm sure that most of you have learned visual discrimination and can distinguish most of these areas, no? Like for example, the presubbiculum, parasubbiculum, entorhinal cortex, and so on. I enjoy particularly sitting down with him and looking at this because with him I learned how cytoarchitectonics can tell you a lot about the different areas of the cortex. And he was so precise, and it was difficult not to be thinking, oh, maybe I'm being biased when I decide these areas, this other area. But the key thing about Ricardo is that he would look at the microscope and he would just see, and whatever it was there, it was there. And it was the rigor, it was just the reality of it. And it didn't matter if it was cytoarchitectonic features or if it was labeled neurons. And if there were neurons, good. If there were no neurons, good too. So one of the first studies that impressed me from when I joined his lab to do my PhD was this work here on the left, where the title of the paper is very telling. It says anatomical basis of memory preservation. And that was because he measured volumes of the entorhinal cortex, and the volumes were measured in postmortem brains, entorhinal, perirenal, and posterior hippocampal cortices, and he found no differences between people who were younger than 70 or older than 70 years old. Sorry. And that was quite amazing. And that's actually the way I got to meet Ricardo, in a talk where he presented his work and a photograph of a section of a human brain that I couldn't find, that he was 107 years old. And I was amazed. So it seemed that the aging could be healthy. Many years later, we've learned, and he has done that too, that there are deposits of very pathological proteins like tau. You can see this in this beautiful immunostaining for tau. And sometimes neurodegeneration happens. And so there was this healthy and unhealthy aging idea that I think still stands up in the literature. So, but as Menno was saying earlier, he started in the trigeminal ganglion and the sensory nucleus of the trigeminal nerve. Just going to show another image that I think this is the type of images that Ricardo would use in his PhD, but this is a clearer one from closer to the time. So I thought I'd show you the trigeminal nucleus. The sensitive nucleus is all these, but this is the main one, and that's what Ricardo studied for his PhD, directed by Don Luis Maria Gonzalo, who passed away, but he was a great wise man in Pamplona at the University of Navarra. And he studied the connections between the sensory nucleus of the trigeminal nerve and the reticular formation. And that gave him, or at least he developed his surgical techniques, because it's not easy to access the brainstem of a mouse, of a rat in this case, put a tracer in there, inject a tracer in there, and let the animal survive. It's very difficult. That type of surgery is incredibly difficult. So he learned all his surgical techniques with these microsurgeries, and he applied that later on in monkeys, as Menno was describing. And when he worked with monkeys, after he went to La Jolla and came back to Pamplona, is when I joined him in the lab. I joined him in 1996. And he was studying the efferent connections of the entorhinal cortex. So this is just two photographs of the monkey brain. This is a rhesus monkey brain. We use the macaque fascicularis, which is slightly different, but very, very similar. So you can see here the entorhinal cortex of the macaque and the perirenal divisions and the posterior hippocampal cortex. And this is the front view with the temporal pole and all the association areas of the temporal lobe. So we place tracers in many, many areas, and then studied the connections from the entorhinal cortex and also the hippocampus. You can see here the dissection of the macaque fascicularis of the hippocampus. Again, one of those beautiful pictures taken by Ricardo, and after a dissection of the hippocampus. And you can see it's interesting to look at this very mini hippocampus. It is 1.5 centimeters compared to the 7.5 centimeters of a human hippocampus. But it allowed us to study the output connections to the cortex from the hippocampus and the entorhinal, perirenal, and posterior hippocampal cortex. So my PhD thesis is one of these arrows, but in the way back to cortex. But all these arrows were traced by Menno, Ricardo, Wendy, Suzuki, David Amaral. So all these summarizes not only the complexity, but also it shows how everything, all the information that is highly processed in cortex is funneled by the perirenal and hippocampal cortex into the entorhinal cortex and the hippocampus. And most of these connections, I would say all of these connections, go back in the same way that they come in. And when I say in the same way, I mean in terms of the density of the projections. And you can see here in this square, this inset here, the thickness of the arrow correlates with, or is related to the density of the projection. But so this is all done in monkeys, as Menno was describing earlier. But Ricardo was always interested in working and translating that into humans. So again, two of these images of the entorhinal cortex of the human brain, and he developed this protocol in 1998 to measure the different subareas, especially the hippocampus and the hippocampal formation in MRI images in humans. So he correlated histology with MRI imaging, something that he kept on doing, and now together with Paul Yushkevich, we've gone to the maximum expression of this work. But he didn't stop in the entorhinal cortex. He also looked at the temporal pole. So he did also a pastillation or a segmentation of the different areas of the temporal pole. He did the medial temporal cortex in MRI images. He also did the piriform cortex and the amygdala. And Laura is expanded our work also in the amygdala, but also the thalamus. So all this work is been trying to apply all the neuroanatomy into the human world. This is just an image showing the divisions that Menno was showing earlier and all the work that went into it. The divisions of the entorhinal cortex, and he had this method of unfolding the cortex into two-dimensional maps, and you can see here in colors all the subdivisions of the entorhinal cortex in humans. And then the picture on the middle, with all the drawings, was a cartoon drawing of not only the entorhinal cortex here in light green, but also all the subareas of the hippocampus and subiculum and perirhinal cortex and posterior hippocampal cortex. And this is an addition or an edited version of a previous one that he had, and that now will be published in the new book of the human nervous system, edited by Paxinos. And here, as I was saying earlier, the beautiful work of Ricardo was just to look on the microscope and identify all these layers that make up all the different subareas of the hippocampus. So, for example, in A here on the an image of these dentine gyres and CA1, CA2, and all the subareas of the medial temporal cortex. And it was very nice to be able to learn to discriminate these then in a larger picture that we have seen earlier with no So this is a larger version of those little squares. So you can see very beautifully the entorhinal cortex in humans with all the layers and the laminate seconds, and the perirhinal cortex area 35 and 36 with the collateral sulcus in the amygdala. And I just thought I'd bring this because he was not only a master of understanding the cytoarchitectonics, but also in preparing these histological sections that were so beautifully done and clear. And then we had on the other side one of the 7 Tesla images that Paul is able to take from postmortem brains and on all his team, and we could use the same cases in MRI images and histology to segment the different subareas, and with hope we could apply these soon automatically to humans, as we heard yesterday from Paul. So this is a slightly more posterior section. You can see still the entorhinal cortex with those clamps that are so characteristic in layer 2 of the entorhinal cortex. The presubiculum, the subiculum, CA1. This is a very difficult area to study because it's as you all know, especially in the hippocampus group, is the uncus, the hippocampal head, and it's extremely difficult to identify all the subareas. But this is CA1, subiculum. This is the amygdala hippocampal transitional area. The amygdala is listed here. And more posteriorly, we have here the dentine gyrus twice because this is still the uncus, and all the subareas, subiculum, presubiculum, and the more posterior part of the entorhinal cortex. And as you all know, when the uncus ends, from here to here, the entorhinal cortex ends as well. And the lateral nucleus of the thalamus is right here, just to help us identify that. So this is just sort of beautiful cytoarchitectonic images that I thought I'd share with you. But not only the cytoarchitectonics were really important, but also the connectivity of the hippocampal formation. And this is a summary that Menno has done earlier in a diagram too. So I'm not going to get into the details, but you can see in red all the inputs from cortex into, if you see at the section of the histology, all in red, all the arrows in red are the inputs from cortex into the entorhinal cortex, and then building up the intrinsic circuitry within the hippocampus, and going back in blue, the projections from the subiculum, CA1 to cortex. And this is also, all these connections are described in detail in this the Human Nervous System book by Paxinos. And we revised them for the new edition. So Ricardo has contributed to this very important textbook, The Human Nervous System by George Paxinos, since the beginnings of the first edition in 1990. And he also contributed with chapters in the Hippocampus book, the 2 editions, with David Amaral on the Neuroanatomy of the hippocampal formation. And a group of us had to take on, for the edition of the Human Nervous System for 2026, and he passed away. So he passed us the work to continue with this Human Nervous System, which is a great honor for all of us. So 1998 arrives, and he moves from Pamplona to Albacete. And I received a phone call from him telling me, I'm going to Albacete, I'll tell you about it. The next phone call was, would you come with me? And I went there to finish my PhD. So you can see us and Mar here in 1998, just when the medical school was starting. And he was then made a professor in anatomy. I completed my PhD, and the medical school started running. And he started the whole department of anatomy. That wasn't really trivial. So his interest, as Menno was saying earlier, was not only monkeys and connectivity and cytoarchitectonics. He also worked with human patients. And one of the, doing the neuroanatomy, of course, of these human patients. One of them was patient EP, who had herpes encephalitis, and you can see here, that destroyed the whole medial temporal cortex. So it was even worse than HM, and it was bilateral. And this patient, even though he lost all the hippocampus and the medial temporal cortex, was able to remember how to go from place to place in Castro Valley in San Francisco, because he was a deliverer. He had a job that allowed him to really know the map. But he moved after deletion into a new neighborhood and couldn't remember anything. But what Ricardo did was to, once the patient passed away, all the tissue was available, and you can see here on the top a control image of the amygdala and entorhinal cortex, and here the hippocampus and the entorhinal cortex, the uncus of the hippocampus, and here on the bottom, the lesion of this patient. There is a documentary about this patient that is actually very interesting to look at. So he also looked at the MRI scans, and of course he did an unfolded map of the cortex of patient EP. So this here is the left hemisphere and the right hemisphere of the control subjects. However, it's a control subject. In the middle, you see a smaller maps where all the medial temporal cortex is missing. And those are the 2 dimensional maps of the patient, EP. So he was able to characterize very precisely what the extent of the lesion was. And that was important to understand patient EP symptoms. But not only did he anatomy, beautiful dissections work with MRIs, but he was very active in society as well. So we can see here he was part of the Alzheimer Association in Albacete. He received prizes, and he was always very engaged into public sort of society and science events. So he would try to inform the public of the importance of body donations and brain donations to science and teaching. So I just took a sample of pictures from news. So he was busy with all the work and all the social engagement work, but he had always time to visit his friends. And so I was very lucky when I was doing my postdocs abroad, he would come and visit me. So he would come to visit me in NIH, and I showed him everything, and we had beautiful conversations also with my colleagues there. And so when I was in London too at UCL. And I just brought one picture from all those stays, and is that when he came to NIH in Bethesda, I was studying then the multimodal nature of episodic memory, and within that context in monkeys, I asked him, could you explain me what's the structure of the uncinate fasciculus? And he said, of course, do you have a brain? we were lucky enough to have a brain, and we dissected the brain, and we could see here so this is a ventral view of the brain. You can see here is the orbitofrontal cortex and the olfactory bulb. You see here the olfactory tract going into the piriform cortex and the entorhinal cortex. This is the lateral, the superior temporal gyrus and the inferior temporal gyrus. This is the temporal pole missing here. And then we showed here, or we opened the uncinate fasciculus, and this is the ventral amygdalo-fugal pathway that would go into the hypothalamus, among other areas, the encephalon. But the uncinate fasciculus communicates the temporal lobe, but also the medial temporal lobe, with frontal cortex. And it does it in a very fun, like a fan fashion way. So when it reaches the frontal lobe, it opens up into different portions. I was interested in the medial portion that is shown in here, that goes into the medial prefrontal cortex. That is very interesting. It was very interesting then for my work, and it's become very interesting afterwards. And also we painted with a pen on the other hemisphere the two areas, and that was very interesting too. So, and then he came to London, where we analyzed also the MRI scans of the patients with developmental amnesia. So these are patients who had hypoxia or ischemia perinatally and some seizures. Those seem to be the two ingredients you must have. And then they lose up to 30% of the volume of the hippocampus bilaterally, and they become amnesic. So they don't remember, in a typical test of memory that you can see here, they don't remember after a delay anything. So he went to Spain and worked in the development of well, he was already working on this now, and the postnatal development of the hippocampal formation to try to understand what was what areas would develop earlier or later within the medial temporal cortex. So just to finish, I will finish very soon. He also studied in the last few years more in detail some sub-areas of the hippocampal formation. One of them was this paper on CA2 of the human, also the gyrus ambiens. And also he worked with Chantal Chavois in Caen, France, and studied this interesting idea of self-awareness in patients who had neurodegenerative disorders, and what could be the networks that hold on to this function of self-awareness of losing memories, or the ability to, it's like meta-memory. How do you recognize your own cognitive abilities? No? That type of self-awareness that is impaired or not, depending on the disease. And so he worked on that, which I think is fascinating. But also we worked on the medial prefrontal cortex, and we segmented these areas in cytoarchitectonics and myeloarchitectonics, and in MRI scans. And we produced these probabilistic maps of the medial prefrontal cortex, trying to understand what are the different functions of the different subdivisions of these medial prefrontal cortex. And we still don't know very much because they're very difficult to understand. So with these, I would like to end by saying that Ricardo started in Pamplona, went to California, came to Albacete, flew all over to visit his friends, and also attracted a huge amount of people in 2023 with the International Hippocampus course. That was a wonderful event. because he brought people from all over the world, including Laura, Paul, and all the lucky people who were in the picture. Most of us are not, but Laura is there, Ricardo is there. In these last few years, he's been collaborating with Paul Yushkevich and Laura and all the Hippocampal segmentation group to try to develop uniform criteria for segmenting the hippocampus. And he also joined me in help me on my own work with human imaging and neuropsychology studies on preterms and long COVID lately. And that's all I wanted to say about Ricardo. There's one more slide just to say thank you, Ricardo, for being my friend, and all of you for your attention. Thank you.
Thank you, Monica. This was really a wonderful overview of a very rich scientific career. This is really great. I think, like I said, we should maybe leave questions until the very end. So I want to remind people that you can put questions in the Q&A box. And what I would propose, which is a bit odd for a webinar, but I would still propose to take a quick break so you can stretch your legs. We find that very important in Sweden to get the blood flowing again. And I would propose to be here in 5 minutes. 4:58. Well, I guess wherever you are, 58, we should restart.
Okay.
This is especially important for Paul. All right, see you in a few minutes. All right, I will slowly start introducing the next speaker as people tune in again. So the next speaker is Professor Paul Yushkevich from the University of Pennsylvania. And he will be talking about Ricardo Insausti's collaboration in computational neuroimaging, building probabilistic atlases of regional neuroanatomy and spread of neurodegenerative pathologies. All right.
Laura,
Please go
thank you
ahead,
very
Paul.
much, Laura and Mark, for organizing this. It's really an honor to present and remember the last 10 years of working with Ricardo. Before I go into the science of the presentation, I just want to tell very briefly the story of meeting Ricardo and working with him for these last 10 years. I actually met him at the very first meeting of the Hippocampal Subfields group in Davis in 2013. But at the time, I'm a kind of shy guy, and I was probably too intimidated to talk to famous neuroanatomists. I don't think we actually interacted much at that meeting, but a few months later, I got my courage together and I wrote him about a possibility of a collaboration. And he answered me with one of the kindest emails I've ever received. First of all, he's very open collaborating, but he also told me that he heard through a friend, Eugenio Iglesias, who I'll mention in a second, that my wife is from Spain, from Andalusia, and he just said, just say hello from another Spaniard, an admirer of her husband. And it's just, I think this email just speaks so much to how Ricardo was and how he could just break down these walls that so many times exist in science between people of different levels and different ranks. And you know, he just treated you with so much kindness and warmth. It's really incredible. I also think it just talks to how much he loved Spain, how much he loved Basque Country. And you know, he's told me numerous times that he had opportunities to work elsewhere, but he very consciously chose to stay in Spain and to build this incredible anatomy department and medical school and train all these people who, many of whom I now have a pleasure of working with. Ricardo invited me to Albacete about 2015. Unfortunately, I don't have any pictures with him there, but I got to see the Museo de Cuchilleria. He took me to the cave houses in Chinchilla, and we had this very interesting experience just on the first day of visiting him. Just somebody, a lady came up to us and asked if we would take the groceries up to her apartment. And of course, she said, yes, let's take the groceries up to her apartment. So on our way to the lab, that's the thing we did. I visited Ricardo along with Eugenio in Pamplona, and got to experience San Fermin, which was his favorite holiday. He always made room in his schedule for San Fermin. Unfortunately, I arrived there too late to see the running of the bulls. And then Ricardo visited us in Philadelphia 5 times, one of those times for a whole month when he came with his daughter, Christina. One time, it was so close to the pandemic that I think if he had taken a flight the following day or the day after, he probably would have gotten stuck here in the US for a few months. So let me move to the science, and I will begin by talking just briefly about this paper that Monica already mentioned, because this work really had a profound influence on neuroimaging research. In the 90s and 2000s, there was a lot of work in computational image analysis on all kinds of advanced registration and segmentation techniques. A lot of the underlying anatomy that was used was quite rudimentary. Many times we would just use segmentation of a single image of who knows where it came from to kind of use as an atlas for segmentation. And Ricardo's 98 paper was just a really amazing example of a crossover between neuroanatomy and neuroimaging, and going in a rigorous way, defining rules in which anatomy could be segmented on MRI, accounting for anatomical variability. At the same time, Ricardo and his colleagues at the Human Neuroanatomy Laboratory in Albacete developed state-of-the-art techniques for brain banking, tissue fixation, and histology techniques that were really optimized for neuroanatomical studies. So as part of this, they performed in situ perfusion so that the fixative could be delivered to the brain right at autopsy and not later when the brain is removed, helping preserve the shape of the brain for neuroanatomical studies. And they really optimized histological techniques so that the quality of the tissue, the ability to distinguish different architectonic areas and cortical layers could be maximized. While mentioning this, I just want to recognize Mercedes Iñiguez, who is the technical laboratory manager who worked with Ricardo for so many years. So much of the data that we've been able to work with is thanks to this incredible quality and art form that goes into getting these beautiful histological sections. So my own work at the time was focused on ex vivo MRI and trying to use computational techniques to combine high-resolution images of the hippocampus into these probabilistic atlases that we could then use to study variability and apply to in vivo imaging. We were working also with histology data and some anatomical annotations, but in a very rudimentary way, not in any of the way that was really optimized for anatomy. When I finally met Ricardo, it turned out that he was already working on something related on a probabilistic atlas of thalamic subnuclei with Eugenio Iglesias, who is a brilliant scientist who's currently associate professor at MGH. And they really did pioneering work on combining serial histological imaging, postmortem MRI, block face imaging, to build a probabilistic atlas of thalamic subdivisions that then could be applied to in vivo imaging. It was later extended to account for diffusion stratigraphy, and eventually, in the last couple of years, Eugenio went on to develop a much larger histological atlas of the whole brain. So there's this whole kind of dimension of Ricardo's work that's incorporated into FreeSurfer that we get to have for posterity. Sorry. So Ricardo and I got excited about mapping out the medial temporal lobe using some of these atlasing techniques. We wrote a grant around 2016, and the primary objectives of this grant were to model the variability of the medial temporal lobe anatomy based on side architecture, then to apply these kind of atlases for automatic segmentation of this anatomy and in vivo MRI, but also to use immunohistochemistry to characterize the distribution and spread of neurodegenerative pathologies in 3 dimensions, and ultimately to develop better biomarkers for in vivo MRI. So the grant was luckily funded, and we developed a very close collaboration where I think both groups build on each other's strengths. We sent hundreds of packages back and forth between Philadelphia and Albacete with tissue and slides, 3D molds. And over the last 10 years, we processed over 120 MTL specimens. In the process of this, Ricardo oversaw the creation of what is probably the world's largest collection of neuroanatomical annotations. So we created this web application in which Ricardo and his colleagues at HNL would go in and label cytoarchitectonic boundaries. And over the years, Ricardo labeled 65 specimens from one end of the MTL to the other. For some of these specimens, he then helped us turn these 2-dimensional annotations into solid 3-dimensional segmentations, as you can see on the right. And then in a series of papers led by Sadhna Ravikumar, who was a PhD student and a postdoc here at Penn, we constructed these atlases of the medial temporal lobe that incorporate his segmentations and use them to study patterns of atrophy and how atrophy is associated with pathology. We have also been working on this goal of extending this to in vivo segmentation, so this can be useful for developing biomarkers and ultimately for studying people living with neurodegenerative disease. And this is ongoing work on validation of this site architectonically informed segmentation atlas for the MTL. Ricardo generated this large collection of annotations, and he always believed in science being open and not being constrained by borders. And so one of the things that we wanted to do is to make these annotations available to the rest of the scientific community. So there's now a website up where you can actually enter and view all of these thousands of annotations, over 8,500 slides and 65 specimens that were generated by Ricardo and his team. And I hope it's just one little way of preserving Ricardo's legacy. Moving on, just going beyond anatomy, part of our collaboration also focused on mapping the spread of neurodegenerative pathologies across the MDL and relating it to atrophy. This is an example from another of Sadhna's recent papers looking at the distribution of neurofibrillary tangles in low and high BRCA stage individuals averaged across a number of specimens, just showing the kind of 3-dimensional pattern of tangle distribution. This is not just purely exploratory, but actually we're trying to leverage all this information we've collected from this postmortem data set for very current problems in Alzheimer's disease. And just to highlight one example, this is our work with colleagues here at Penn to study mismatch between tau pathology and atrophy, and in the process of this mismatch to discover biomarkers or patterns on MRI that can distinguish presence of co-pathologies. And this is particularly looking at late, which is a TDP43 co-pathology, and showing that there are distinct patterns that can, if we look at the patterns of mismatch, we can detect the presence of late with pretty high accuracy. 0.9 if we look at postmortem MRI, which of course we cannot do in living patients, but also not so bad 0.8 in antemortem MRI. So in if this translates to living patients, that's a pretty good accuracy of detection of late co-pathology. So I want to close to say that this collaboration is never going to be the same without Ricardo, but I'm very happy to say that we are continuing to work actively together. This picture on the top left is just from yesterday when we had our monthly meeting with the UCLM and UPenn teams, and we have quite a few goals in mind. We have been collaborating on grants, and we hope to keep going for as long as we can. And I think, again, this is probably the best way we have to honor Ricardo's memory and just keep all this stuff that he started. So just want to say thank you to Ricardo for being an incredible role model, and thank you to Ricardo's family for sharing him with us. Thank you all for listening.
Thank you, Paul. That was a really nice overview, and it's also really nice to see all the histology work that he and the team at UCLM did. That it is publicly available. It's already available now, right? Like people can access it. Yes.
Yes. But wait till the end of the meeting.
Yes, don't be distracted. All right. Then it's my turn next. I will share my slides. All right, so I will be presenting the last talk on behalf of the Hippocampal Subfields group, which, as was already mentioned today, is working towards a harmonized segmentation protocol for medial temporal lobe subregions. And as was mentioned, Ricardo has actually been part of this from the beginning, from the very earliest meeting in 2013. And I was smiling when Paul said this, but like Paul, I was actually too impressed with him to go out to talk to him. And little did I know that 2 years later, I would actually be visiting Alpha Theta and visit Ricardo and the rest of the team, where I had an extremely wonderful visit. It was so welcoming. They took me out for dinner every night for a week, I think. And we started a very long, a 10-year-long collaboration, mostly on the amygdala that I will not be talking about, but the work that will hopefully be submitted later this year. Yes, I will continue with the hippocampal subfields group. So I guess I don't have to tell you this, but the medial temporal lobe, as we've heard in the last talks, is really crucial for cognition, aging, and neurological and neuropsychiatric diseases. And as we've seen, it consists of several subregions, which have differential functional roles and also differential vulnerability to disease. As Paul already mentioned, and in this figure, you can see that different MTL subregions show different vulnerability to neurodegenerative diseases, the topic that I'm particularly interested in, where you can see that darker colors are associated with a higher pathology load. And we can actually use imaging to pinpoint these early neurodegenerative disease processes, for example. So what you see here on the right is the famous figure from Braak and Braak that showed the accumulation of neurofibrillary tangle pathology in transentorhinal region or Brodmann area 35. It's one of the hallmarks of Alzheimer's disease. And you can see on the left that that is also the place where we see very early changes on the MRI scan. Similarly, with functional imaging, we can pinpoint functional differentiation. And on the right here, you can see a very seminal paper that showed functional activation in specific hippocampal regions during a memory task. So for those of you not so familiar, how do we label subregions on MRI? So basically you can scroll through the brain from anterior to posterior, from the front to the back. And on each slice, you can outline or label or color the regions of interest. And if you put them all together, you get a 3D rendering. As you see on the right, you can measure volume or thickness or surface deformation, whatever you want. So on MRI, especially with these beautiful T2-weighted MRI scans, we can see some of the borders that are of interest. I hope you can see my mouse. So you can see a dark band in the hippocampus, but we don't see all of the borders. So for example, the border between pink and red, we cannot see. So what we do is we look at anatomical atlases and we try to come up with the best approximation of where to place this border. Unfortunately, this is not bulletproof, as you will see in our next slide, but I think this is the best common practice. And fortunately, there are a lot of automated methods now that can automatically label this. Whereas I did most of my PhD, I labeled this all manually. I did, I think, hundreds of scans like this. But as I already alluded to, there's not one labeling protocol, but actually there are many different segmentation protocols, over 25, and I think it's still going up. And what you see here is an example of that. So this is one slice in the same brain that is labeled by different laboratories. And you can see that, of course, in this brain, the different regions, the different colors should all be in the same place, but it's not the case. So for example, if you really focus on the pink region, the perirhinal cortex, you can see that it covers a different area for each of the 3 laboratories. So if each of these laboratories would do a study on the perirhinal cortex, you wouldn't know how to compare these results. So that's very confusing. Another example to really explain why this is so confusing regards to aging. And this is based on a review by De Flores et al., which investigated or kind of summarized the body of research that was done associating aging with volumes in the hippocampus. And there were 18 studies. You see each of them here. One figure represents one study. And the colored regions reflect the regions that show a significant association with aging. And you would assume aging is one thing. You would assume all of these studies would find the same result. But in fact, you see that any constellation of significant associations was reported. And while, of course, this could be due to study sample selection, a big issue is, in fact, we think, the different labeling schemes. And what you see here initially is that I labeled all of these little hippocampi the same way, but in reality, it looks more something like this. And I always like to highlight these 2 studies. The study by Lachois et al. Looked at aging and hippocampal subfields and found an association with subiculum volume. I myself did something similar, but then found an association with the subregion CA1. And while these labels are different, if you look at these 2 pictures, you can see that there actually is a big overlap. So it's not clear if we find a different result, if we find a similar result, but just a different label. And this is exactly the problem that we're trying to solve. So in 2013, we started an international group of researchers who collaboratively developed a harmonized protocol for segmenting MTL subregions on T2-weighted MRI. And as I mentioned already, Ricardo has been really integral to this effort from the very beginning. The idea for this protocol is that it's validated, it's reliable, and we can apply it to any population imaginable. And we have been lucky to have been funded by the NIH since 2022, which really helped move this project along. It's a really large effort, over 200 members. It's still growing, it's still open, so I want to highlight that. For those of you who are not part of the hippocampal subfields group, feel free to join. There's definitely a lot of nice opportunities for early career researchers. So I won't have time to go into all the details and all the results, but I want to kind of highlight our general approach. And actually, our general approach starts with collecting labeled histology. And the reason we do this is because anatomical references have actually been showing different findings, different places where borders between regions are located, and we wanted to get to the bottom of this. So we have been extremely lucky to have been working with 4 amazing neuroanatomists, including Ricardo, of course, who have been extremely generous with their time and their resources. They have been creating these labeled histology sections for us, which we can then use to kind of come up with rules for the MRI. But throughout the years, they've also been generous in answering all of our questions regarding anatomy, which has been really instrumental for our progress. So with these this histology data, we've then worked on developing subfield boundaries on high resolution MRI, so rules for that, develop a protocol. Whenever we have this, we do a feasibility check. We collect feedback from the larger hippocampal subfields group to make sure that we didn't overlook anything, that people are all on board with the protocol. And then we do a formal reliability analysis to make sure that if you apply this protocol twice, you get the same result. So we've made a tremendous amount of effort or progress over the last few years. So we've divided the whole MTL into 4 regions. We started with the middle part of the hippocampus. We always say it's the easiest part. It is, but it's relatively speaking, it's still rather complicated. And we have been able to follow all these steps, and the paper was published, I think, 2 weeks ago. So this is really fresh from the press. The next thing we did was we started working on the anterior part of the hippocampus. That is still in progress, but we're making a lot of good progress, and hopefully we will be able to finish this soon. The posterior part, the tail of the hippocampus, was finished, and it's submitted and available as a bioRxiv preprint. So if you're interested in that, please check it out. And then we're also working on the MTL cortex. And a big part of this effort has been to work in groups. We've done a lot online, but we've also had the opportunity to host a lot of these meetings in person, which has been instrumental. We've been working in groups to come up with rules, to understand the anatomy, etc. And as you can see in some of these pictures, Ricardo has been really kind enough to join us in these meetings and to help with the protocol development. And I think he also really enjoyed being part of this community. I wanted to highlight one specific meeting in Albacete, which was mentioned already earlier, which Ricardo had actually been asking for several years if we can please have a hippocampal subfields group meeting in Albacete. And we finally managed to do this in 2023, where a large number of actually early career researchers had the amazing opportunity to take a 2-day neuroanatomy course on this section and learn. And it's been really an amazing experience. I also wanted to take the opportunity to highlight our upcoming meeting in Bordeaux, which will not be on protocol development, but here we will actually teach people the new parts of the protocol that are already done. So you can, I think, I'm not sure registration open, but keep your eye out for the registration, which I think will open soon. So before I end, I also wanted to point you to some resources. Ricardo has been part of our community for a very long time, and he's been part of several of these recordings that are placed on YouTube, one webinar, a podcast, but also the several parts of the Albacete meeting were recorded and are available to watch online to learn more about anatomy. And I think it's really amazing that we are able to preserve this for future generations to learn about neuroanatomy from the best, I would say. Yeah, so that's all I had to say. I want to thank you for listening and for joining in today. And yeah, of course, thank Ricardo for all his input and help with this important effort.